Hi, my name is Pierre Champetier. I'm a postdoctoral fellow at the Paris Brain Institute in France, studying the sleep of elderly individuals. But why study sleep in this population? Well, let me share an anecdote. So, this is me on holidays at my grandparents' house, and I remember that my grandpa used to ask me each morning if I had slept well. So I usually replied yes, but when I asked him the same question, he often replied, not that good. I woke up several times and my sleep was very light. In fact, this anecdote is quite common, as half of elderly people complain about their sleep, and this number is even higher in individuals starting to develop Alzheimer's disease, the most common neurodegenerative disorder, which notably affect memory. And that's a big issue, as today we know that sleep disturbances can be a risk factor for developing such terrible neurodegenerative disorder. Moreover, there is currently no cure available to this disease, while the United Nations projections foresee a surge of Alzheimer's cases due to the increasing aging of the global population. So better understand which aspects of sleep are impaired in the aging population is crucial for potentially delaying the onset of the disease. To do this, we have to measure sleep. How can we measure sleep? Well, we can use this. So this sensor is a kind of antenna to record electric activity of your body. If I put such antenna on your scalp or on your face around your eyes, for instance, I will be able to record your brain activity and your eye movements even if you close your eyes. Based on such measures, we know that during sleep we alternate between two different sleep phases called non-REM and REM sleep. While keeping your eyelids closed, your eyes move during part of sleep called REM sleep for rapid eye movement sleep, but they stay still in non-REM sleep for non-rapid eye movement sleep. The brain activity also differs between these two sleep phases. So if you imagine that your brain is a gigantic orchestra, during REM sleep your brain cell would play music at different rapid rhythm, while in non-REM sleep they would synchronize to play together at slower rhythm. Thus, while sleeping, our brain switches between two different sleep phases, as if the brain cells were playing two different sleep melodies, non-REM and REM sleep melodies. Unfortunately, these melodies are altered with age. Notably, the brain cells are less synchronized during non-REM sleep, as if the music score was blurred. The two sleep melodies are even more altered in patients with Alzheimer's disease, especially the one in REM sleep, as if the overall music score was even more difficult to read. Interestingly, some researchers have recently focused their attention to a very specific instrument of the non-REM sleep melody. If we listen to this instrument, we would hear something like this. So in this audio, each beep represents a sleep spindle. A spindle is a burst of activity generated by the brain that we can record with our electric antenna on someone in non-REM sleep. And recently, researchers noticed that spindles are not homogeneously distributed. Some are isolated, while others are gathered together. More precisely, there is a rhythmicity with irregular alternations between periods with few isolated spindles and periods with several gathered spindles, alike verses and chorus within the melody of non-REM sleep. Similarly, during REM sleep, eye movements are not homogeneously distributed. There are alternations between periods without eye movements and periods with several gathered eye movements, also creating verses and chorus within the melody of REM sleep. Importantly, an increasing number of research suggests that the structure of non-REM and REM sleep melodies with such verses and chorus is important for memory consolidation and sleep stability. However, these studies have been performed on mice and young adults, while little is known in the aging and Alzheimer's disease context. So during my PhD, I gave the very first evidence suggesting that the structure of spindle versus and chorus during non-REM sleep is impaired in older adults, leading to a more homogeneous non-REM sleep melody. Only a few studies have investigated versus and chorus of the melody of REM sleep in this population, and no consensus has emerged. Moreover, the two sleep melodies still need to be explored in patients with Alzheimer's disease. And this is exactly what I want to do, thanks to the Brainstorm Neuroscience Pitch competition. This is indeed an amazing opportunity for me not only to share my work with a broader audience, but also to continue my research and start my own project that will explore for the first time the verses and chorus of sleep melodies in patients with Alzheimer's disease. For instance, I want to know if the verses and chorus of non-REM and REM sleep melodies undergo specific alterations with Alzheimer's disease, and if such alterations could contribute to memory issues in this population. So to do so, we will recruit 3 groups of participants: young adults, healthy elderly, and patients with Alzheimer's disease. We will monitor the sleep during one night in the lab, and I will perform a memory task before and after sleep. We will compare the structure of the non-REM and REM sleep melodies, which we will relate to their memory consolidation as a memory task. So by diving into the vaults of non-REM and REM sleep, we might identify key sleep parameters impaired in Alzheimer's disease, encouraging other research groups to also quantify these sleep features. In the middle term, such sleep modifications could represent simple and valuable sleep features constituting a cue for questioning an upcoming Alzheimer's disease. In the long term, these discoveries might contribute developing strategies to restore sleep melodies and sleep quality in older adults and patients. And who knows, maybe when I will be old, to my grandchild question, "And you, Grandpa, did you sleep well?" I will answer, "Yeah, not that bad." Thank you very much.